Lecture 13, and this is actually going over pre-transfusion case studies. This is actually your assignment 2. So if you guys have already looked at that and have questions, let's talk about those questions now. If you guys haven't looked at this, we're going to go over the case studies, and then if you guys have questions from now until Friday, I'm happy to help answer those questions. Okay. So this is your assignment 2. All right, assignment 2, you have a panel and you're given some information, okay? So you're given a little patient history here. This panel was run on a pregnant female. She had 2 previous pregnancies. What does that tell us? She's been exposed to foreign antigens. She has the potential for antibodies, okay? IgG antibodies, all right? She's O negative and her IAT screen was positive, hence we're doing her panel, okay? Do you guys have any negative cells that you're able to rule out anything? Just one, right? Only panel cell 8 is your negative cell, and it's negative across the board, all right? Remember, if you have a positive reaction in any phase, you can't rule out with that cell, okay? So this is our only cell that you're allowed to rule out with on this one. So that makes it very challenging for blood bank, right? How can we rule out anything, identify the antibodies, and identify any underlying antibodies? What antibody does it look like based on your pattern of reactivity? It could be P1, but notice your initial spin reaction, P1 would be reacting, would be negative with panel cell 1. It would also be positive. Where did I see that at? 2, yeah. Notice that P1 is on panel cell 2, and we have a negative reaction at initial spin there. All right, so it doesn't really match our pattern of reactivity. Ah, okay, so Lewis B, notice Lewis B is on panel cell 3, 4, 5, 6, 7, and then 10 and 11. What else? That doesn't explain panel cell 1 or panel cell, was that 9? Panel cell 9. Could she have both? She had both Lewis A and Lewis B. Yes, she could. All right, so you have Lewis A reacting at initial spin in 37. You have Lewis B only reacting at initial spin. Does that make sense to everybody? Does everybody see that? All right, but what else do you have? You have some AHG reactions going on here too, right? And they're 3+, so they're very strong. So did any antigen present on cells 2 and 8 and 11? Cal, right? So, but how can we prove that Cal is there? How can we get rid of these Lewis reactions? Neutralization, okay? But remember, when you do neutralization, even though these reactions are going to go away, you still have to honor that patient's antibody. Okay, so we've established this patient has anti-Lewis A and B. Now we're trying to see what else is under that Lewis A and Lewis B. All right, we need some negative reactions for ruling out. Okay. So then, after Lewis neutralization, notice that our initial spin reactions are now negative. The reactions that were at 37 are now negative. And our AHG reactions are still there, okay? And so we can rule out based on these reactions, and indeed, we do have Cal there, okay? Now, I want you guys to make sure you understand when you guys do Lewis neutralization, you are completely repeating the panel, all right? So you are treating patient plasma with your manufactured soluble antigen, your Lewis substrate. right? Has a soluble antigen in it. It will absorb out the Lewis antibodies out of your patient plasma. Okay, that's why they don't react, because they have been absorbed out of your patient plasma. So you treat your patient plasma, you take that treated or neutralized patient plasma, and then you repeat the panel, right? So you'll have to use a whole new, it's the same, you can use the same panel, but you have to repeat it, okay? Does that make sense to everybody? Okay. So you could do it on a new panel sheet, and then you could redo your crossing off. Sarah asked the question, do you still need to do select cells that are Lewis A, B, and Kale negative? I don't think you need to do select cells in this case, because after the Lewis neutralization, the only thing reacting is Kale. Everything else is ruled out. And then Lewis A and Lewis B are there. And let me see, you do have, this is a tricky question, because you remember the rule of 3, you have to have 3 positives and 3 negatives. Okay, so if you break this down on this particular case, notice that if you just look at Lewis A, notice that you have panel cell 1 and panel cell 9, you have 2 positives there, right? That are only reacting that initial spin in 37, okay? So if you had to follow the rule of 3, you would need to run an additional Lewis A positive cell to get that rule of 3. Okay. And then if you look at Lewis B, you have, and Lewis B is reacting at initial spin only, right? So you definitely have 3 positives. And let me see, you do have, and if you've established that Lewis B is only reacting at initial spin, you do still only have 2 negative cells. So you probably need to run some more negative cells. Does that make sense? To follow the rule of 3. But you don't need any more select cells, all right? You would only need to run more cells to get the rule of 3. Okay. But everything is ruled out, and if you were just trying to reach that 95% confidence level, then you would want to run some more cells. So some of the questions that are associated with this, would you want to phenotype the patient? What would you phenotype for? Kale. So there's a big debate on whether you should phenotype this patient for Lewis A, Lewis B. What phenotype must be present in order to build Lewis antibodies? She has to be Lewis A negative and Lewis B negative. And do you guys remember anything about the Lewis antigens during pregnancy? The antibody is IgM, so it can't cross the placenta, therefore it's not really a concern in causing hemolytic disease of the newborn for the fetus. But for the antigen expression, remember your Lewis A and Lewis B is on the oligosaccharide of your red cells, okay? They are known to have a decreased antigenic expression during pregnancy. So you will see this frequently in blood bank. You will see pregnant females that have either Lewis A or Lewis B or a combination of both. Okay, you will see that in pregnant females. And that's just because the Lewis phenotype is known to decrease during pregnancy. So my point with that is there's controversy on whether you should phenotype for Lewis A and Lewis B.
Is it decreased to such a degree that they can possibly build autoantibodies to their Lewis?
It is decreased to the point where they could have an antibody. Yeah. And that's typically why in pregnant females you will, and especially in the African American population, you will see anti-Lewis A or B in pregnant females. Yeah. Now, again, that usually that antibody will typically go away after the pregnancy. So what are your concerns for the fetus? Which one of those antibodies should we be concerned about? Kale. Right, so Kale is IgG. It is the second most antigenic antigen. It's second after D, right? So we definitely want to make sure that we give her Kale negative units, right? Oftentimes, because Lewis A and Lewis B are not associated with crossing the placenta, causing hemolytic disease of the newborn, most clinical blood banks are not going to phenotype for Lewis A and Lewis B, and they're not going to, if that patient needs a transfusion, they're just going to give cross-match compatible. They're not going to worry about phenotyping the donor units either, right? There's some controversies over it. If you're working in the blood bank and your SOP says to phenotype, then you have to follow the SOP. But AABB, FDA, all of those standards say that you can just give cross-match compatible. You don't have to phenotype, okay? All right, that's case number 1. Anybody have any questions? Yes, you would definitely want to phenotype for Kale. There's controversy on whether you would phenotype for Lewis A and Lewis B. So. This is everything that we just talked about. The Lewis family is also associated with hemolysis. It is one of our antibodies because it is associated with the IgM. It is very efficient at activating complement. And you guys remember when complement is activated, it can cause hemolysis, okay? Any clues with case 1 that were helpful for you? Number 1, she's pregnant, right? What's this patient's likely race? African American. 22% African Americans are the Lewis A negative, Lewis B negative phenotype. All right, in your clinical implications, Kale is capable for causing hemolytic disease of the newborn. Lewis A and Lewis B are often not so much the concern. If the patient needs a transfusion, what type of units will you guys give her? Kale negative blood. This one says Lewis A negative. If you were to look at your panel, because Lewis A was reacting at 37 degrees, right? So if you think about 37 degrees is the temperature of your body, then you might want to honor that Lewis A. So it just depends on what your SOP requirements say. Now, if you were to give her, because you're going to do your cross match at 37 degrees, if you were to pick your units and they were compatible at 37 degrees, that would be efficient, okay? That would satisfy the requirement. All right, so here's your next panel. This is case number 2. You have a pregnant 25-year-old female. She has had a previous pregnancy. And your reactions here. So if you do your ruling out, you see that we still have possible big E, little c, and Kale that are not ruled out. So based on your pattern of reactivity, which one makes sense? Little c is definitely there, but you can't rule out big E, right? And then also Kale. So what would you do in this situation? You need to definitely run some select cells. Try to rule out that big E. It's going to be very difficult. And then, but definitely try to get some little c negative, Kale positive to run as your select cells. Okay. Were you given a, you don't have any phenotype, okay. Oh, your genotype, right? So if she has anti-big E and little c, what's her possible genotype? Yeah, so she could be R1, or she could have big D, big C, little e. She could be homozygous for that. Or she could be, remember, we don't know her D phenotype, right? So this is why you have two possibilities. Right? So she could have the D antigen expressed on both genes, or she might only have that D expressed on one gene. Does that make sense? But we know if she has the big E and little c, she must be little c negative and big E negative. Okay. So if you were to convert this to your Weiner nomenclature, what would it be? What is this? R1. What's this one? R1. Little r prime. Okay, or you could say r prime if you want. Prime goes with your little r language. Okay, so R1, little r prime. All right, so what's your most probable racial origin of a donor with the following typing results? So we have Lewis A negative, Lewis B negative, Duffy A negative, B negative, JS is in your Kale family, positive for A and B. What population do you typically see A negative, B negative in, Lewis? African American. Is that the same for your Duffys? Does anybody remember why? Malaria. Remember, your Duffy antigen is thought to be a receptor site for the malaria organism. So your malaria can bind to the red cell through your Duffy antigen, and that's how it enters the red cell. That's how the infection occurs. So if you lack the Duffy A and B antigen, there's no receptor site for the malaria to enter the cell. So it's thought that the Duffy A negative, B negative is a built-in defense mechanism against malaria. So that's why we typically see it in the African American population. Okay, so this patient is probably African American. Oops. All right, so what about the Kale antigen? What do we know about him? What are some characteristics of him? It's the second most antigenic. All right, so it's very strong, second only to D, like I mentioned earlier. Sarah had a question. Sarah asked if that's supposed to be JK. I think it's supposed to be JS. JSA, JSB is in your Kale family. Most of your population is going to be positive. They're public antigens. Most of your population is going to be JSA positive, JSB positive. All right, so if you look at cell number 6, what is your Rh genotype? So cell number 6, do you think it could be R1? If you're looking at cell number 6, can it be R1? Remember, if it's R1, that means it has the D antigen. Okay, so cell number 6 cannot be R1. Right? Could it be little r, little r1, little r prime? Could it be little r double prime? Yes. Right. Remember, the double prime indicates that the big E antigen is present. Okay, and that you're negative for the C antigen. So you could have little r double prime. You could also have, we have little c being expressed and little e being expressed. So you could have, what is that? R to the y, right? So it would be like this. Your patient, where did the eraser go?
It's over by the hand sanitizer.
Oh. We're looking at cell 6. Right here. All right, so your patient is D negative, big C negative, big E negative, little c positive, little e positive. Okay? Given that this patient is D negative, is the D gene being expressed at all? No. All right, so we know his phenotype has to be this. Okay? Can he be little prime? Can he be prime? Because he's big C negative, single prime. No, all right. Could he be double prime? No, all right. So he has to be this, right? Let's see. Does that make sense? And that converts to what? Little r, little r. Does that make sense to everybody? Okay, so that's your Fisher race and Weiner nomenclature. Okay. Did you get that, Sarah? You got it? Okay. Okay. That's totally opposite from what that says. What cell are they looking at?
I think it might be referring to the actual question on the solo.
Oh, it provides a table. Oh, it does. My bad. Okay, so I was using 6 from the panel. All right, So you have to make sure you pay attention to the results that you're giving, all right? So look, oh my gosh. Somebody should have stopped me. Okay. So in the example that they give, you could be, if you're D positive, we don't know if both genes are expressing the D or only one gene is expressing the D, right? So that's why they could be R1 or little r. But what's your other possibility? If they're R1, but that example has you as little c positive too, right? So you have to have that little c antigen being expressed as well. And they're big E negative, so both genes have to have the little e being expressed. Does that make sense to everybody? That's totally confusing. Okay, I messed that up.
Can you go over that again?
Okay, so what I was doing, I didn't read the question that was in the case studies. So what we originally established was for this cell. Okay, the actual question gives you a phenotype. All right, so what's your probable Rh genotype for this? All right, D positive, but do you know if both genes are expressing the D antigen? You don't, all right? So he could potentially be homozygous or heterozygous, essentially. But remember, there's no little D antigen, it's an amorph, all right? So we know that big C is being expressed, but big E is not. So he could be R1 on one allele, and then on the other one, he could be little r because little c positive, E positive. So both of those antigens have to be present. Does that make sense? So here's your possible combinations. He could be both D. So that's your question, the D antigen, all right? Is he homozygous, only one gene expressing it, or both genes expressing it? That's what we don't know. We can't tell from our blood bank testing. Okay, so he could be, but because he's big C positive, little c positive, his genotype must include both of those. All right, so you have big C, little c, big C, little c. He's big E negative, so none of your possibilities could include big E. Does that make sense? Raina? And then you have another one, you have for number 7, you're given another example. Notice in that one, he's positive for all of the Rh antigens. So notice if you're positive for all of the Rh antigens, you're D positive, big C positive, little E positive, and big E positive, you have to have all of your antigens being expressed. The thing is, the reason you have different scenarios is because of the variation in the D. With our blood bank testing, we don't know if the D antigen is being expressed on both genes or if only one of them. So that's why you have 2 different possibilities. Does that make sense? Because even if only one gene is expressed in the D antigen, he's still testing Rh positive with our serological techniques. that make sense to everybody? All right, so question number 8, ask about your week D. What's your week D test? Yes, remember your week D test is kind of like a additional step in your RH test. All right, you definitely want to add AHG. Remember that it doesn't become week D test until you get to the AHG phase of testing. Okay. What's the purpose of that 37-degree incubation? That's because remember, most of these antibodies react at 37 or AHG. So by incubating at 37, we are affecting the temperature, which might enhance the antibody attachment to the antigen on the red cells. Therefore, it might enhance agglutination. Right? So it mimics what goes on inside the body. Okay? So that's the purpose of the 37. Right, number 9 is you're giving a mother and father, so it's a little bit of a paternity test. What's the genotype of your offspring? Could you have an offspring that's R1, R1 from those? No, you couldn't. Could you have one that is little r prime, little r, meaning that big C is present? Notice that the father is the only one that has the one gene that has the D antigen with the big C antigen, okay? So you're probably not going to have a little r prime allele, okay? That's probably not possible, or it's not possible. Could you have one that's little r, little r? That's possible from your mom and dad, right? So if you look at your question, this is actually not an option in one of your answer choices. All right, so even though this is a possibility, it's not one of your answer choices. So this one is. Little r, little r. All right, so here's one that's asking, oh, yes, ma'am.
Question, I think it was 8 up there with the last slide. Just to be clear, so the 37 degrees is to express the curve. And then the in vivo at the AHG.
Yes, so the purpose of the 37-degree incubation per week for your D testing is to allow that antibody to uptake onto the cell. Remember, because we're testing for D, so what characteristics of our D antigen? They can react at 37 and AHG, right? So that's why we include that 37 degree. And remember, if you think about the purpose of that test, the procedure of that test, you incubate, spin, and read. Did that incubation at 37 enhance that antibody onto the red cell membrane? So that's why we look for agglutination again, before you wash and add AHG. right, so this is just a genotype question. Possible Rh genotypes. Could the offspring be R1, R1? Yes, they could. Could be R1, R1. Could they be R1 little r? Yes. So question 11 asks, which of the following red cell typing most commonly found in the Black donor population? What do you guys think about that one? Definitely A negative, B negative. And then number 12 is another probable genotype. What do you guys think about that one? D positive, big C positive, big E negative, little c positive, little e positive. Notice that the only negative Rh antigen there is big E. So is A an option? Is B an option? What about C? Why is C not an option? Big C is not being expressed in that one, right? Is D an option? Yes, because you have big C, little c, and little e being expressed in that genotype. All right, question 13, you're given some patient typing results. Negative with anti-A, negative with anti-B, 2+ reaction with your A, B, that's your forward typing, and then your reverse typing, 2+ with A1 red cells, 4+ with B cells. Your antibody screening is negative. What do you guys think about that one? Why do you have a negative reaction in your forward typing with anti-A, but a positive reaction with A, B? It's a subgroup of A. What do we know about subgroups of A? They have very weak antigenic expressions, correct? That's why we have a negative reaction with our anti-A. And then in your reverse typing, you have an unexpected reaction with the A1 cells. What does that mean? They have anti-A1. Question 14 says you have a patient that has the genotype little h. So what does that mean? They're Bombay, meaning that they have the production of no H substance. Okay, remember, in order for the A and B antigen to be added onto the oligosaccharide, you must have H substance, which adds the L-fucose sugar to the oligosaccharide. All right. So even though this patient's genotype is AB, what's our forward typing going to look like? The patient's going to be O. Even though they have the A and B antigen, because of the lack of that L-fucose, that A and B antigen is not able to add on to the red cell membrane. Okay. So with our blood bank serological testing, our patient is going to be type O. We're almost done. Okay. So question 15, you have a female admitted with anemia. Blood samples received for cross-match, 6 units red cells. Patient is group A, Rh negative, has no history of transfusion or pregnancy, but the following results were obtained. If you look at the results, screening cell 1 is positive in the IAT at AHG phase of testing 3+. Screening cell 2 is also, and the auto control is positive at IAT testing, AHG test. And then all 6 donors are also positive in the cross match. What do you guys think? What's going on there? What do you think about that? Huh? What do you guys think about that auto control being positive? It's an auto antibody. So what would need to be done? Remember, saline replacement is for rouleau. How about an auto absorption? What's the auto absorption going to do? So it's an absorption. It's going to try to absorb out whatever that antibody is in the patient plasma. So if you adsorb it out, then we could repeat your IAT and hopefully it would be negative, right? So we could try to do a adsorption. So depending on what phase of testing will determine if it's a warm or a cold. Typically, if it's a cold autoantibody, you could try to identify the specificity of it. Like, is it anti-I? Is it a P1? Is it that kind? So then we know what type of cold antibody it is. With warm autos, it just reacts with everything. There's really, sometimes warm autos can take on the specificity of little e. We'll talk about that later on. But usually with warm autos, and even after an absorption treatment, sometimes it doesn't even go negative. All right, so patient number 16, we have a paternity investigation, produces the following phenotyping results. Father is type B, mother is type O. Can the child be type O? Yes, all right, so we can exclude the father based on ABO. Could you exclude the patient or the father based on RH typing? So the father is RH positive, mother is RH positive, big E positive for the dad. What do you guys think? The child, is the child big E positive? No, big E negative. So we exclude the father based on that. All right, and then the last question, patient's forward typing, you have 4+ with anti-A, negative with anti-B. In your back type, you have an unexpected reaction with your A1 cells. You have a 1+ reaction and a 4+ reaction with your B cells. What do you guys think about that? What's going on there? Could be anti-A1. What else could it be? Could be some type of cold reacting antibody as well. What next step would you want to do when you suspect a subgroup of A? A1 lectin and A2 cells. We're not given that result, but we are given the screening cells. So you have your IAT results. Screening cell 1, screening cell 2, and your auto control are all positive at initial spin, indicating that it is a cold reacting antibody, auto antibody, because your auto control is positive. Does that make sense to everybody? Okay. All right, that's the last one.
Real quick, back to the previous question. The
Uh-huh.
one.
Yep.
Since we're only given, I guess,
Yeah.
are we assuming that that's homozygous?
I'm assuming since you're not given it,
We're not giving the other one?
that she's negative, that they would be negative. So like the mom would be big C negative, little e negative. Oh, sorry, that's the dad. I was looking at the dad. And the mom would be little c negative, big E negative. Because since they're not, I'm assuming since they're not listed, that means they lack that antigen. All right, you guys let